Okay, tools of molecular genetics. This is just a brief presentation just to go over some of the testing methods that we use just so we can review it. So a mutation is a change in DNA which affects the function or translation to the protein. So anything that changes the DNA code that results in either abnormal or absence of the end product, which is a protein, is a problem. So changing in the DNA can do a lot of things. One is, first we make a copy of this that goes out into the endoplasmic reticulum. It's used as a template to make a protein. And there are a lot of things that can go wrong. And we're going to go over a lot of these over the course of the next 2 years. But one is a point mutation. So if the sequence is supposed to be ATG, then mRNA is U, because A goes with U, T goes with A, G goes with C. And this puts into the protein a tyrosine. But what if we go from ATG to ATA? Then we get UAU, which is also a tyrosine. So this is called a silent change. It doesn't really affect anything. Yes, there's a change from a G to an A, but we still get a tyrosine, so who cares? But what if we go to ATC? Well, then we get UAG, which is a stop codon. So wherever this is, it just stops, and we get really no protein. Or we change from an ATG to a GTG, which puts in a histidine instead of a tyrosine. And that's a big deal. So the third base is known as the wobble base, and sometimes if you change from this to this, you still end up with the same amino acid. Sometimes, though, you end up with the worst possible thing. So it kind of depends on where the protein is, like where this amino acid is in the protein can also determine if it's a problem. Probably one of my biggest issues now is as we do more genetics testing, there's 3 possible answers when getting a genetic test report back. One is normal, and this would be considered normal. It wouldn't even be in the report. One will be definitely pathogenic. If we see a stop codon, it's going to be pathogenic. And then my all-time favorite is, gee, there's a variant of unknown clinical significance, which means I'm not really sure. And so that comes into pass of how we manage the patient. So let's go with the easiest thing, which is sickle cell anemia, because everybody has the same change in the base pair in sickle cell anemia, so it doesn't cause me headaches. So it goes from a GAG to a GTG. That results, instead of glutamic acid, valine. Every single person with sickle cell anemia has the same change. Now, one of the ways that we can determine if there's a change in the DNA sequence is to cut the DNA with a restriction enzyme. And this notices something called a palindrome. It's the same sequence going forward and backwards, and it will cut at that region. There are over 3,500 restriction enzymes. So we can choose one that cuts where we're interested in. And we're going to talk about this with sickle cell in a minute. But let's just talk about the different ways to look for gene function. And one is a southern blot. This actually looks at the DNA in the nucleus. Is the DNA correct? A northern blot looks at, is the RNA correct? Northern blots are used a lot in cancer because certain cancer genes are amplified, and so there's a lot more RNA, so we can figure out how much there is. And a western blot looks at the end product, which is the protein. We don't use these blots as much as we used to because they're expensive and time-consuming, and we have better methods. But I'm just going over this, so you may not want to spend a lot of time remembering this, but you cut your DNA with restriction enzymes, you separate their size, you then transfer them onto a filter paper after you've used your agarose gel to separate them. And we used to use fluorescent radioactive tags. We now use fluorescent tags to figure out where it migrated to. With a northern blot, again, the same thing. We extract RNA, we separate it, we transfer it, and then we see where it is. And here we have a western blot. We actually take the protein and we separate it. We're going to talk about Duchenne's muscular dystrophy in the spring, but this is a muscular disorder where the protein dystrophin isn't formed correctly. So here is normal dystrophin. And Becker's is a milder form that you get dystrophin, but you can see it's a little bit shorter. So when we look at the protein, it's not the same size. Whereas in Duchenne's, you get no protein. You don't get this band here. And the dystrophin gene is a very large gene, and we used to have to do this analysis on Western. We now are capable of sequencing the gene, so I don't use a Western anymore. But as I promised, we're going to go back to sickle cell, and we're going to talk about sickle cell anemia and how we use restriction enzymes to determine if somebody is affected, carrier, or doesn't have the gene. So we use an enzyme to cut this little fragment out in the middle, and then it will also cut right here at the at the GAG, which is the normal allele. But when it changes to CTG, the restriction enzyme doesn't cut it. So we get, in patients with sickle cell, we get one fragment, a larger fragment. In individuals with normal hemoglobin, we get two fragments. So in the sickle cell, because of this change, they don't cut right here. So if you have just two small fragments, that means you have normal hemoglobin. If you have just a big fragment, that means you have just sickle cell hemoglobin. And if you have both, you're a carrier. So in this family history, these parents have normal hemoglobin and the sickle because it didn't cut here, both of them. In this child, they gave both normal copies of the hemoglobin. In this child, they gave both sickle cell, and in this child, they gave normal and sickle cell. So this is a carrier, affected, and not even a carrier. Now, PCR, or polymerase chain reaction, is a faster method, especially if we have a few or one mutation. And what PCR does is you need a very small amount of DNA, and you can look at the area that you're interested in and amplify using polymerase enzymes that DNA into larger amounts that we can study. So we have our DNA, we make it single-stranded, we have a template, we amplify our DNA using our polymerase, and here you can see in just a few amplification sequences, we can end up with a great amount of DNA, and this allows us to use a cheek swab or just a small amount of DNA to do this. And this is actually the method that they use in newborn screening, which in Illinois we actually test for sickle cell anemia by newborn screening. And so they can have a primer for normal hemoglobin, they can have a primer for the sickle cell hemoglobin. And so if you amplify both normal and sickle, then you're a carrier. Only sickle, you got sickle cell, and only normal, you're good. So the next thing I want to talk about is Sanger sequencing. And this is how we sequence a gene. This is the gold standard. It's incredibly expensive. We don't use it as much anymore because we have easier and faster methods. But I just want to talk about how it's done and why it's such a great way to do it. Now we know what the sequences of the gene were, but we used to not. So if we had a fragment of DNA, this was many people's PhD projects for like 30 years, that they'd pick a gene or sequence of DNA, and that would be their project. They have a fragment of DNA, and they're trying to figure out what the sequence is. So they add their fragment of DNA into 4 different solutions, and in each one of these solutions, they have the regular base pairs ATCG, and in this one they have deoxythymine, deoxycytosine, deoxyadenine, and deoxyguanine. When the deoxy base is added, it stops the fragment from continuing because it stops it from continuing to sequence. So in this first fragment, it put C, A, G, T. It picked the normal T this time. A, T, picked the normal T, C, C, A, C, and then it put the deoxy in there. But in this fragment, the first time it put the normal T, but the second time it put the deoxy. And then the third time it put the deoxy right at the beginning. So we have a bunch of fragments, and then we separate them. And we do the same thing for all of these. And then when we separate these fragments, we can then say, oh, the shortest is, we got a T, an A, a T, a C, C, A, C, T, G. We can sequence this fragment doing this. You can see, though, this is a pretty expensive and time-consuming method to sequence a gene, but this is actually how we sequence, like, what is the normal sequence of the gene? Because we need to know what's normal before we can use better, cheaper, easier methods to figure out the rest of it. So cystic fibrosis, there's over 1,400 known mutations, probably more than that, for the CFTR gene. Some occur only in certain families, and these include missense, frame shift, splicing, and nonsense mutations. But Delta F508 is the most common. 70% of patients with CF have actually this one mutation, especially in Northern Europeans. And so this is a little bit different of a carrier frequency than the one I showed you in the Mendelian, because sometimes they say it's 1 in 25, sometimes they say it's 1 in 29. But I just want to kind of go over this as far as mutation testing and what that means if you have carrier testing. So if we look for individuals of Ashkenazi Jewish descent, 94% detection rate. If we do mutation testing, so the prior risk is 1 in 24, but it goes to 1 in 400, so it like really drops a lot. Whereas Caucasians, it's only 88%, so we drop half the amount, just this 94 to 88% detection rate. Hispanics, only 72% detection rate. So the prior risk is 1 in 46, only drops to 1 in 64. So here we have a double risk to those of Ashkenazi Jewish descent before you took testing, but it's higher after testing because the testing isn't very good. And when you get to Asian Americans, what's the use of even doing carrier testing, because you haven't dropped your risk all that much. So one of the things when we're asking if you had carrier testing, what's your prior risk and what's your after risk? Like, how much did you really drop your risk? Because if you don't have a very good detection rate, you might not be dropping the risk that you actually carry this. So for CF, screening panels are available, and again, negative testing will reduce the risk, but it doesn't completely rule it out. So one of the things, if one parent is a known carrier for cystic fibrosis, carrier screening is not good enough. We actually do Sanger sequencing and deletion duplication. We actually sequence the other parent's genes. We just don't do the run-of-the-mill, let's look at the common mutations. Let's do better. Especially, let's say there's a Caucasian parent that's a carrier of CF and the other parent is Oriental. You do not want to do common carrier screening because you're not going to pick up that much. So mutation screening benefits. It's quicker, cheaper. You can look for common mutations when we're doing population screening. A lot of times when somebody's pregnant, they do carrier screening. And if they're a carrier for something, then they really look carefully at the other spouse. But if they're not a carrier, then we don't necessarily look at the other spouse. So mutation screening limitations, it doesn't tell you for rare mutations. So rare disorders or rare mutations within a common disorder, if you test negative, you haven't completely ruled out the chance that you're going to be a carrier of this. And one of the examples I'd like to use is Tay-Sachs is a disease common in Ashkenazi Jewish population. And yet nowadays, for probably 3 generations, people of Jewish descent have been tested. So most babies that are actually born with Tay-Sachs are not Jewish. So that concludes this lecture.